Welcome to my presentation on functional food attributes of selected underutilized plants. I am Nazrim Marikkar, currently work as head of the food chemistry program at National Institute of Fundamental Studies. So in this seminar presentation today, I would like to discuss with you 3 of my projects currently ongoing at the Institute. First one is nutritional studies on Terminalia catapa. Second one is anti-diabetic potential of underutilized leafy plants. Third project is stevia rhodiana as a non-caloric sweetener. First of all, we would like to focus on project 1. So in this, we are going to discuss the Terminalia catapa as an underutilized plant. This plant is widely distributed in Sri Lanka, and it sheds thousands of metric tons of fruits annually, which goes waste due to without any usage or any value addition. Currently in Sri Lanka, there are 2 different varieties of Terminalia catapa exist. The first one is the purple variety, and the second one is the yellow variety. So the Terminalia catapa belongs to the family Combretaceae. So this slide shows the visual appearance of the fruits from the 2 varieties. The first one is the purple variety shown in the above, and usually below we find the yellow variety, and usually we cut open the fruit and subjected the fruits into dehydration, and dehydrated seed can be cracked open, and we can recover seed kernel from these fruits. And this can be before research investigation, this one can be dehydrated and stored in the refrigerator for further analysis. So in this project, we have 3 different focus. The first one is intervarietal comparison of proximate composition, and the second focus is intervarietal comparison of oil extraction and characterization. Third one is intervarietal differences in biological activities and functional food attributes. First of all, we would like to show you the results obtained from intervarietal comparison of proximate composition. So we have measured the parameters like moisture, protein, fat, ash content, and carbohydrate content. Based on the results, we have identified that fat component dominates, followed by protein component. So in both varieties, 60 to 66% of fat occurs, while both varieties contain 25% of protein. So the yellow variety the oil content of the yellow variety is dominating, and while the oil content of the purple variety is 60%. So as the oil content is exceeding 40%, we can subject the seed kernel into oil extraction using an oil expeller like this. We call mechanical oil expeller, so which is free of any chemicals, additives, or any solvents. Without the help of any solvents, we can isolate pure oil using this oil expeller. So for this, we dry the cotamba seed kernel, and we can feed into the machine, and through this exercise, we can collect about 33% oil and about 66% defatted residue. So our project aim is how to use this oil and the defatted residue for value addition and value enhancement. So when we analyze the oil composition, the fatty acid composition of the oil, so we found out the most dominant fatty acid is palmitic acid, followed by oleic acid and linoleic acid. Oleic and linoleic occurs almost equal percentage, and the linoleic acid is the functional essential fatty acid that occurring in the seeds, and its amount is 28 to 29%. So as you know, the alpha linoleic acid is an essential omega-3 fatty acid. It supports heart health by lowering LDL cholesterol. It supports brain health and cognitive function. It possesses anti-inflammatory properties. It improves skin health. It supports blood sugar regulation. All these are functional properties of alpha linoleic acid. After this, we focus on the mineral distribution. So potassium, calcium, magnesium are the most dominant major mineral components present in the oil. So out of these three, potassium dominates, followed by calcium and magnesium. So these three minerals are important for the overall health. Particularly potassium supports heart health, calcium supports bone and teeth health, and magnesium also support various metabolic processes occurring in our body. After the major minerals, we come across minor minerals in the seed kernel, and zinc, copper, and manganese are the most important, most dominating minerals in the seeds. And zinc is very, very important for overall health of our body. Likewise, copper and manganese also play their own roles in the metabolic process in our body. So after discussing these two focuses, and now we are coming to the third focus, functional attributes of Terminalia catappa seed kernel. And it has nutraceutical properties, medicinal properties, and various other functional attributes, which we want to discuss it in further. So under this category, we investigate total phenolic content, antioxidant activity, anti-obesity activity, and anti-hyperglycemic activity. These properties we are comparing for both purple and yellow cultivar, and for that matter, we take the extracts of the seed kernel using hexane, ethyl acetate, and methanol. As you know, hexane is the non-polar component, and methanol is the most polar component, and ethyl acetate is in between the two. So after taking the crude extracts through the sequential extraction of the seed kernel, so we found out the highest TPC, total phenolic content, was found in methanolic extract of purple cultivar. So the highest DPPH activity was detected in hexane extract of yellow cultivar. So here we use ascorbic acid as the control. Then we investigated ABTS radical scavenging activity of the three extracts, hexane, ethyl acetate, and methanol. And the highest ABTS activity was detected in methanol extract of purple cultivar. Then lastly, we under antioxidant activities, we investigate the FRAP reducing power. So out of the three extracts, only methanol extract shows the highest activity, and the best activity was detected in purple cultivar. So after that, we investigated the alpha glucosidase activity of the 3 extracts, and the highest activity was detected in ethyl acetate extract of purple cultivar. So here we have used acarbose as the positive control. As the project outcome, one article got published in the Journal of Agricultural Sciences Sri Lanka. This was published last year. Now I want to give a focus to the next project, project 2, functional attributes of 5 underutilized plant species of Sri Lanka. Here we have investigated Licolapala, Copacola, Stevia robertiana, Yakinaran, and Korakaha. These are the 5 underutilized leafy plants we have investigated. So before we investigating the functional properties of these leaves, we have powdered the leaf sample after drying, and we have taken sequential extraction using hexane, ethyl acetate, and methanol as solvent, and we have collected crude extracts. And after getting the crude extract, we have subjected these crude extract into analysis, total phenolic content, antioxidant activities, anti-hyperglycemic activities, and anti-obesity activity. So out of the 3 different extracts, we have found out the highest TPC content was found in Yakinaran. So we have 3 extracts: hexane extract, ethyl extract, and methanol extract. Among the hexane extract, Yakinaran one was found to show the highest TPC content. So secondly, we have investigated the ethyl acetate extract. In this also, the highest TPC content was detected in Yakinaran. Thirdly, we again investigated the TPC content of methanol extract, and in this one, Licolapala was found to contain the highest TPC content among the 5 different leaf extracts. After determining TPC of the plant sources, we now move on to investigating the antioxidant capacity of these plant extracts. So again, I would like to remind you. So we have 5 plants, and each and every plant was subjected to hexane, ethyl acetate, and methanol. So it is a sequential extraction process. So first we focus on hexane extract. Among the DPPH radical scanning activity, the highest activity was detected for ethyl acetate extract of Yakinaran. When coming to ABTS radical scavenging activity, the highest activity was detected for methanol extract of Licolapala. So when coming to FRAP antioxidant power, the highest activity was detected for methanol extract of Licolapala. So after investigating the antioxidant activities, we now move on to alpha glucosidase inhibitory activity. The first one is we have checked all the 3 extracts for this alpha glucosidase inhibitory activity. The highest activity was detected for hexane extract of Yakinaran. So among the alpha amylase inhibitory activity, the highest activity was detected for methanol extract of Licolapala. We also investigated lipase inhibitory activity of the plant extract, the highest lipase inhibitory activity was detected for methanol extract of Licolapala. As the project outcome, one article got published in Food Chemistry Advances. It was published last year. Now I would like to focus my attention to the project 3, Stevia rhabodiana as a non-caloric sweetener. So this plant is very much important, especially when we consider about a growing number of chronic diseases like diabetes. It is a small herbaceous perennial plant which can be grown under the climatic condition in Sri Lanka. The active ingredient in these leaves is steviol glycoside, which is responsible for the sweetness of these leaves. Actually speaking, the sweetness of these leaves is about 100 to 300 times higher than sweetness of normal sugar. So in this project, we have given focus to 3 important aspects. The first one is stevia vegetative propagation. Second one is stevia syrup development. Third one is stevia crystal development. And let us see these 3 different aspects one by one. First one is stevia rhabodiana vegetative propagation. And we are collecting stevia leaf cuttings, and from that we can collect stem cuttings. So stem cuttings are used for vegetative propagation, and in this one we are selecting the best soil mixture for the healthy growth of stevia rhabodiana, and we are mixing sand soil and topsoil in 1:1 ratio. And from this mixture, base mixture, so we are mixing this base mixture with cow dung, cocoa peat, poultry litter, and biocompost. Out of these 4 treatments, base mixture with cow dung gives the best results, and a large number of survival was observed to be under this condition. And after the propagation, we are transplanting these newly produced saplings into pots, and from that, so we can grow it under the open sky or greenhouse. So we have tested their growth under greenhouse as well as open sky. The best results were obtained for the open sky treatment. So this is the vegetative propagation part of this plant. After that, we are moving on to producing a syrup which can be used as a sweetener in place of fermentable sugars like sucrose. So we have 2 different series of the syrup development mixtures, and top side we are showing syrup mixtures prepared from fresh leaves, and down series shows the syrup development by using dry powder. So we tested both fresh leaves as well as dry powder. So we want to see which one is going to work better for us. So out of these 2 series, the one that was obtained using dry powder was found to be the best. After that, we have checked how much solvent and how much plant material need to be used. Here we are taking edible grade ethanol as the solvent, and we are mixing with water. So 10% ethanol, water, first one. Second one is 30% ethanol, water. Third one is 50% ethanol, water. Fourth one is 70% ethanol, water. Finally, right, 90% ethanol, water. The other one is 100% ethanol. So using these extracting medium series, we have obtained syrup from the fresh leaves as well as from dry powder. And we have tested how much of material need to be mixed with the solvent. And we have tested two levels. One is 1 gram is to 25 gram milliliter solvent. The other one is 1 gram is to 50 milliliter solvent. So out of these two, we have found out 1 gram sample material mixed with 50 ml extracting medium was the best for our purpose. And this variable concentration ensures that which one is the best. And for that, so we have checked through sensory evaluation and what is the best condition for extraction. So we have tested 10% mixture for 24 hours, and we have tested the 30% mixture for 10 hours. And lastly, we have checked 50% mixture 9 hours. So these three levels of solvent mixtures were subjected to extraction for variable time period, 24 hours, 10 hours, 9 hours. So the whole different formulation types were cross-checked using sensory evaluation. And we found that the best results were obtained for 30% extracting medium solvent mixture for 10 hours will be the optimum condition to get the best syrup out of stevia leaf dry powder. Lastly, we focused our attention to recover steviol glycoside from these leaves. And I told you that the sweetness coming from the stevia leaf is due to steviol glycoside, that is the active ingredient. And we are subjecting these fresh leaves into dry powder. And the dry powder then subjected to boiling with water, and we are getting water extracts. And it is a turbid solution, and therefore we add a little about a calcium carbonate treatment. Then after adding calcium carbonate, we are getting a dark chocolate color solution. And we want to purify this solution using activated charcoal. So in order to purify, we added a limited amount of activated charcoal into the solution so that we can recover dark-free solution, which was when it was subjected to crystallization, we were able to recover steviol crystals as shown in this slide. So our results of our investigation shows that from 1 kilogram of dry powder, we were able to recover 80 grams of stevia sugar. So leaf into water, 1:10 ratio we have to take. Temperature, we need to maintain about 40 degrees Celsius for 4 hours for crystallization, so that we are in a position to collect steviol crystals which can be used as a sugar substitute for diabetic patients. So as the project outcome, we were able to obtain one article published in Ceylon Journal of Science. And also the student who worked in this project was honored with the gold medal during a convocation at the Sabaragama University of Sri Lanka, and she received the gold medal from the Vice Chancellor at the convocation ceremony. Finally, I would like to express my sincere thanks to two of my research assistants. First one is Ms. Savani Ulpathakumura, and second one is Fatima Fahbida. Both of them untiringly worked in this project, and which gave a lot of results for us. And then I would like to acknowledge the funder, the National Institute of Fundamental Studies. They have given generous funding to carry out these 3 projects. With that, I would like to express my sincere thanks to the Institute for providing me necessary facilities and funding to carry out these nationally important projects for Sri Lanka. Thank you.